Nasinov algorithm is a base pair maximization algorithm which starts with small subsequences and then extends in order to capture the larger sequences. Here is the algorithm. We see that there are 4 different scenarios or cases. You can have a matched pair which is being added into an already matched pair I plus 1 and j minus 1. So this I and j is actually, it's a lingo over here because match over here means pairing. So it's not the exact match, it's whether we have G or C, whether we have A or U. So in that case they are pairing with one another. So if they pair up with one another, we will have a good weight or a high score. Other situation is where we can have an unpaired element which is being added. In case 2 and 3, or in the 4 case, we are looking into a bifurcation. So let's fill up a matrix. So we have a sequence G G G A A A U C C. So actually same sequence on both the axes. We are starting with the terminal nucleotides and we are trying to just match them or find the pairs starting from one terminal, and we are trying to pair it up with the other terminal. So in this way, on one side we will move inside out, and on the other side we are moving outside in. So one of them, j goes to like j minus 1, j minus 2 and so on, and I goes like I plus 1 and so on. So we do those steps, dynamic programming steps. So there is initialization, matrix filling and traceback. So first we initialize 2 diagonals which are arrays to 0. Diagonals, here we are not looking for something which is matched to itself. So we initialize, or these diagonals are 0, so we are not interested in that. So actually we are interested in this is upper half of this matrix, or we can take the lower half. So obviously it's kind of duplication on itself. So we are actually not interested on the diagonals, but we are looking for pairwise comparisons between for each nucleotide position with every other nucleotide position within the same sequence. So we fill in squares and we sweep diagonally. So here we start, for example, we can see that these bases, we have C and C on both sides. They do not pair with one another. So in this way we can say that it's unmatched pair, so our score stays as 0. Remember, on one side we started with the last position. We started with, for example, J on one side, and on other side we are starting with I minus 1. As we move up, it becomes I minus 2, and on other side we are looking into the next position on J. So we keep on doing, and in the end we can come fill up this matrix. Let's focus on one cell in for instance, and this last one, how we fill up this matrix, and you can do some practice in your homeworks and fill your own matrices where you can understand how it works. So right now we are at this position. We have this terminal C which we are comparing with the first nucleotide, which is actually I. So there is dynamic programming. So the possible path is we can get that score from this j minus 1, whereas it was 2. So it's coming from I and j minus 1. The score was 2. So we can fill it up as 2. Either we can get this as 3 from this bottom cell, which is highlighted with this pink color. So right now we are where we are getting this score from I plus 1 and j. The third scenario is we can get the score from the diagonal, where we can have I plus 1 and j minus 1, and we can add up plus 1 since it's a diagonal move. So in this way our score, then which one is giving us the best score? So obviously in all 3 scenarios which we have seen earlier, we will get the best score from this 3 bottom one down there, this one. So we then keep our trace back pointers as we did in our multiple sequence in our pairwise sequence alignments of where we were doing those PSA problems, and then we can move along with those our pointers, and we can trace our structure. So the structure becomes like this, where we can look into these different bifurcation cases where we can have our reference coming from those diagonals. So we can have a bifurcation, we have maximum score coming with this equation down there. So this base pair maximization will not necessarily lead to the most stable structure, which it might create structure. Since it's looking into just base pairing, we might end up with thermodynamically unstable structures. So that is the drawback for this technique. So it's comparable to aligning sequences with scattered matches. So maybe we might end up with some sequences or some structures which are not biologically reasonable.